So our first speaker is Donald Stein, who's a professor in emergency medicine at Emory University. And you might think, well, what's this got to do with a professor of emergency medicine? But he's an expert on brain plasticity, and you're going to hear about that and its relation to progesterone. And I want you to give him a special applause, because you'll see that Donald is mature. He's getting on in years. He's perhaps almost as old as me. And he's just written his first blog, and he's feeling very excited about it. So I'm sure he'll promote that, and I hope you'll all go and read his blog after you've heard his speech. Donald.
Thank you for inviting me. You know, this is the second or third time I've been back, and I've had the pleasure and the honor to teach here, and it's always a great pleasure for me to be back and to see my friend Verity and others. It's just a wonderful place, and you ought to be very proud to be here. So I'm going to start this talk and give you basically a 28-year journey into if I can find it. I'm a Mac person, so I have no clue how to get this up. Oh, there it is. I see it. Thank you. Okay, thanks. Now I'm set. So I'm going to talk about a 28-year journey of how we came to think about progesterone as a potential treatment for brain injury. Most people think, what? A female hormone? A hormone involved in birth control, maybe? A hormone involved in development? How could that possibly be available for something like the treatment of brain injury? Well, it's taken a long time. I always like to start with a disclosure, basically that along with Emory University, we are a party to a licensing agreement with BHR Pharma, a small privately held French-Belgian company that actually makes progesterone. And although none of the research that I actually do has been supported by BHR, I have consulted with them, and we do retain royalty rights to some of the development that goes on. The university gets the vast share of everything, but I needed to disclose this to you. I also want to thank everybody, and one of you is right here in the audience, Justice Cowles, who worked with us. It's been a long story. This is not the size of my lab. This represents all the people over all the years that made the story possible. It's a great group of people, and they're just put up with a lot of abuse, a lot of doubt that progesterone could be a successful treatment for brain injury. So it took a lot of courage, I think, for many of these students and postdocs to start out on a career where the trajectory was far from certain when we began this journey so long ago. What's the unmet need? Most of you may not know this, but traumatic brain injury is also a very significant public health problem. There are no available clinical treatments right now for TBI in close to 50 years of research. Like with other areas you've heard about, most of the pharmaceutical companies have pulled away from this area because so little has been effective. TBI affects the entire spectrum of development from infancy to old age, and there are no neuroprotective treatments coming along. So TBI is the problem. Just in the United States alone, there are about 52 to 60,000 deaths a year, over almost 1.5 million hospitalizations due to traumatic brain injury. 300,000 of those occurring in people your age or much younger. Pediatric TBI is a big, big problem. In fact, it kills more young men under the age of 35 than all other diseases combined. Mostly men, by the way. At least 3 TBIs are sustained every minute. Males are 3 times more likely to die of TBI than females. I'll tell you why this is the case, but it's in general due to males are stupider and they put themselves in more difficult situations, as you know. But there's other good, really, really good reasons for this. So, one of the big problems we're facing right now in the United States is that our involvement in these two wars in Afghanistan and Iraq have produced over 250,000 traumatic brain injuries in young men and women. And the Veterans Administration and the government is overwhelmed with trying to provide effective treatments because there is no early stage. So most of what has to happen is these people go into long-term inpatient or outpatient care. It's a huge expense. over $70 billion a year now. It's been a big, big problem, but thanks, in a way, thanks is the wrong word to use, but because of the tremendous involvement with the military right now, there's more attention being paid to finding suitable and effective treatments for both pediatric and sports injuries. Just to give you some idea of what's going on, if you look at multiple sclerosis, spinal cord injury, HIV/AIDS, breast cancer, and traumatic brain injury, TBI far exceeds the numbers of people suffering from these disorders. Spinal cord injuries, if you look at that, until very recently, the entire budget of the NIH was identical for research for traumatic brain injury as it is for spinal cord. That was about $90 million a year. Now, because of the military involvement, it's substantially higher. So I won't dwell on a lot of the statistics, but one of the things I do want to point out is why have there been so many failures? Why have we been so unsuccessful in finding an effective treatment? Partly because I think it's the way in which people characterize traumatic brain injury. It's a systemic disease, as is stroke. When you have a traumatic brain injury, even if it's a relatively mild traumatic brain injury, it's going to produce changes in virtually every organ of your body: your heart, your lungs, your liver, your spleen, your gut. And if you're looking to target a specific gene or a specific receptor or a specific site of in the brain where the injury might have occurred, the amygdala, the hippocampus, the cortex, you name it, you're very likely to fail because you're not dealing with the systemic aspects of the disease process itself. When you look at this and you can see that what TBI does, it produces changes in all of these characteristics. I'm not going to go into any of this because it's not the purpose. I want to tell you how we move from clinical trial. But all of these events take place within the first minutes to hours after a traumatic brain injury. And all of these events have to be stopped, and neuroprotection has to be added on if you're going to find a way to stem that cascade of biochemical and morphological events that lead to destruction of nerve tissue and alter glial neural responses. Those are the support cells of the brain. This process can unfold days, weeks, months, years after the initial injury, as we're finding with people, for example, who suffer mild repetitive concussions or due to improvised explosive devices in Iraq and Afghanistan, and then months to years later develop post-traumatic stress disorder, which many people still think, unfortunately, many psychiatrists still think that this is a case of malingering or other types of psychological behaviors, as opposed to those due to the metabolic and biochemical changes that are often not seen with traditional imaging techniques that need much more sophisticated analysis than is currently available. But that story is beginning to be told. So recently, clinical trials have been very disappointing. There have been over 50 compounds tried. Every single one of them now has failed, including hypothermia, acetylcholine, hypertonic saline. You can read this list very well, and you can see for yourself. I need not repeat it. Every single trial has been a disappointment thus far. And again, I want to stress that the reason for this is because if you're looking to target one neurotransmitter or one receptor, it's almost a foregone conclusion you're going to fail when you're dealing with a systemic complex disorder like traumatic brain injury or stroke. So a lot of people recognize that traumatic brain injury or stroke is just the beginning of the disease, not the end. It's just the beginning of what has to be dealt with from both an early acute stage treatment as well as long-term rehabilitation that also needs to be factored into this, and all the other changes in plasticity that need to be dealt with that are both positive as well as negative. Plasticity doesn't always represent something that's beneficial. You can get reorganization of the cortex after a traumatic injury that takes place within minutes to seconds, and that can lead to lifelong disability because it's stamping in the wrong behavioral and functional mechanisms. And how do you overcome that? These are all issues that need to be studied. Again, it's a systematic complex disease. Look at this. I just want to show you something. If any of you ever go to the Society for Neuroscience, you'll see 37,000 people presenting 22,000 posters, none of which have any relationship to one another. And most of it, it's true. I was a founding member. I say this with a certain degree of sadness. But you look, this is just one pathway for one signaling of NF-kappa B, which is a factor that's involved in the brain injury process. And all these signaling pathways, if you want to write them down, I'll leave it on for another second. But you can see that this is a very, very complicated story. And if you're looking for one pathway and one signaling pathway, and this is just one for one protein, you can begin to get how complex this story is. So progesterone and brain injury. I'm not going to read all of the slides here, but this is a very important one by a good colleague of mine who spent her life studying progesterone receptors and pathways in the brain, Roberta Britton. She's at the University of Southern California. And she said that most people think that progestins, as I said, target the uterus, but they also affect every other major organ system, including brain, cardiovascular system, immune system, and the generation of blood cells. This is going to become important to the progesterone story, and you'll see why. Progestins interact with many receptors, including estrogen, androgen, glucocorticoid, and mineralocorticoid receptors. So progesterone and its associated family of progestins can affect a number of different systems. The receptors are abundant in areas of the brain involved in reproduction, as well as everywhere else in the brain. This is important. You'll see later. And these molecules regulate aspects of brain function, from how brain cells connect to one another. Think about that if you're thinking about repair. And as well as to the birth of new neurons, and how these also affect mood and cognition may be discussed by some of my colleagues. But the point is, one of the things that I think why progesterone, and this is the take-home message, I'll tell you now in case I run out of time, progesterone is what's called a pleiotropic hormone. It acts at multiple receptors, multiple genes, multiple pathways, not just the classical progesterone intranuclear receptor, but a wide variety of others. And we're discovering more and more of these mechanisms and pathways as we go forward with our research. So how did I get to progesterone and TBI? It's an interesting story. When I was much younger, back 28, 30 years ago, there were reports, and even earlier than that, anecdotal reports in the clinical literature that females recover better from TBI and trauma and stroke than males. This was very controversial until recently, when a retrospective study going back and looking at the American National Trauma Data Registry with 1.8 million subjects, they found that when they went and looked at this, there's no doubt now that in evaluating over 1.5 million cases of trauma, that women clearly recover better than men in all forms of trauma, not just brain injury. But as part of this, when I was back beginning this study, we saw these reports that women tend to recover better than stroke and trauma. And I said, well, has anybody looked at this in the lab? And at the time, no. There was a couple of women, one of them very famous, a woman from McGill by the name of Doreen Kimura, who happened to be beginning way back to look at this. Most of her stuff was ignored because it didn't seem particularly relevant. But we asked whether this could be replicated in rats. And we began observing sex differences in rats, and this is all published data. I'm going to give you my website if I have time. My new website. You heard about my blog. You can go look on the website and find all kinds of interesting stuff. My students put this together, by the way. I'm very proud of them. Female rats had less functional deficits than males, with similar injuries. These were very large frontal cortex injuries. Female rats showed substantially less evidence of brain swelling than males. And what was so interesting about this was that the outcome of the work in females depended on where they were in their estrous cycle, the equivalent of the human menstrual cycle, at the time the injury was inflicted. And basically what we found was that when we looked at the brains of these animals, there was virtually no swelling and much better functional recovery when the animals were injured when they're in their cycle, their estrous cycle, when they were high in progesterone as opposed to high in estrogen. And that's what led us to all of the work we did. Again, we weren't giving anything at the time. We were just looking at what happened. Subsequently, we began to look at whether we could treat with progesterone. I said, well, let's see what happens if we gave progesterone. And at the time I started this, back in the 1980s, people said to me, you're nuts. You can't give a female hormone to males. It'll never work. It'll cause all kinds of problems. It's really bad for you. The difference is that unlike estrogen, progesterone doesn't produce any changes in secondary sexual characteristics. So we began to give injections beginning always after the injuries occurred. And these were almost complete removals of the frontal cortex in the rats. What we found was that after a very short course of treatment, anywhere from 3 to 5 days, that's it, we found marked recovery of function on a variety of behavioral tests. All of this is published, and you can see, go to PubMed or go online or go to our website, and you'll see all these articles for yourself. We were dramatically able to reduce inflammation, programmed cell death caused apoptosis. The size of the injuries was reduced anywhere from 50 to 70%, and we got dramatic improvements in functional outcome. Most importantly, for clinicians, you could delay this treatment, and this will become important when I tell you about our clinical trials. You can delay treatment up to 24 hours and still get really beneficial effects. That's one of the few drugs that I'm aware of that has this capacity to produce major functional recovery, even after a delay of 24 hours. I would never recommend waiting just 24 hours, but this becomes very important in field treatment, if you think about it. What's interesting, we found that aged rats, we looked at the developmental spectrum, aged rats we also respond to treatment, but we require higher doses. We found that tapering the dose works much better because you don't get inflammatory rebound. And what's a nice side effect or benefit is that progesterone's metabolite, allopregnanolone, also works very, very well in the treatment of stroke and traumatic brain injury. And we've been studying all these different pathways. In ischemic stroke, it's also a big problem. And there is no effective treatment for stroke. There's one treatment now called tissue plasminogen activated, TPA. It's a clot buster. It breaks up clots. The problem with TPA is that if you give it after 3.5 hours, you can cause a hemorrhagic conversion and the patient can bleed out. So only 3 to 5% of all stroke victims ever get TPA. It's just too dangerous to give, and it's extremely expensive. It costs about $4,000 per treatment. That's one of the downsides of progesterone, actually, I'll mention now, is that it's so cheap that not one major pharmaceutical company ever expressed any interest in moving forward with this because you can't patent it, and it's dirt cheap to give and administer. It's an interesting conundrum to deal with. So stroke, as you can see, is also a big issue. It's interesting that stroke ischemic events are more frequent in men. Women are less likely to have a stroke in early and mid-adulthood than men. Stroke incidence doesn't equalize in women and men until they're over 75. Women actually respond better to TPA treatment, and there are just many differences, again, between women seeming to do better. So all this seems to fit. If you look at some interesting data in pregnant females when they're in their third trimester, that's when progesterone levels are very high. Progesterone levels in a female in her third trimester are about anywhere from 30 to 50 times higher than when she's not pregnant. And when you look at the third trimester of just before birth, there's virtually no incidence of any kind of stroke. Whereas just around delivery and afterwards, when progesterone drops precipitously, there's a marked increase in stroke incidence. It's interesting, with stroke, we're getting similar beneficial effects. You can see a marked reduction in the size of injury. That's important. The ischemic injury is much smaller, meaning you're damaging less cortical tissue. You can delay up to 6 hours. This is very, very important because ischemic stroke, if it isn't treated within the golden hour, the chances of a successful repair is very limited. This window of treatment, without giving TPA and seeing a 50 to 70% reduction in the size of the infarct, again, all this is published, is pretty dramatic and very unique. And you also show not just a reduction of infarct and reduction of physiological injury, so to speak, but you're also seeing marked improvements in cognitive outcome. And we know the mechanisms. I don't have time because I thought I was going to speak to you for 2 hours and have a private seminar, but I'm just going to zip through the fact that we know a lot about the mechanisms. And if you go on my website or write to me, we can tell you a lot about this. It has a lot of effects. You can look at all of them here. It does a lot of different things. That's the main take-home message for you. If you want more details, you go on PubMed, you write to me, you look at the website. All this information will be there for you. It does a lot of different molecular things, mechanisms that reduce inflammation, improvement in water transmission. It affects glia. It stimulates glia to make more myelin. It does a lot of beneficial things at the same time that it's blocking a lot of the cytotoxic events that often follow a traumatic brain injury or stroke. The main point to remember, and I can go on and on about this, it does a lot of different things. It works at a lot of different receptor sites. It's not just limited to one receptor pathway. It's not just limited to one protein marker. It has widespread effects. And I'm going to mention this now why it's important, because if you think about progesterone as a developmental hormone that's evolved to protect the fetus, you got to think, why does this hormone, not estrogen, not testosterone, why does progesterone increase 50-fold in a pregnant female and stay that high all throughout gestation and then drop right after birth? This is a hormone that's evolved to protect the fetus. And the processes of development, I've written about this long before any of you were born, that the processes of development are very, very similar to the processes of repair, or vice versa. So it's not a particularly brilliant hormone. It's just doing very similar things during development, or during repair and regeneration as it does during development. And again, a lot of this is written about. So quickly summarizing, it does all these different things. Everything, that first balloon chart I showed you before, one of the things we found is that basically progesterone affects every one of those, that cascade of biochemical toxicity in beneficial ways. It either enhances trophic factors, it blocks excitotoxicity, it improves mitochondrial function, it reduces the cell death. It does a lot of good things. And again, I'm rushing because I only have a short time to tell you about it. It affects a lot of different genes. We've looked at all this. We continue to examine it. We know what many, not all, but many of the actual genes that get expressed or inhibited as a result of this progesterone treatment. So I can't talk to you about all of them, but I assure you that it's all in the literature. And when you look at this is an interesting chart that's come out in systems biology. Every time you have a disease, you're not just affecting one disease network. That's what this message shows you. You're affecting all kinds of, if you have a neurological disease, you can follow these colors. It's going to affect all these other gene networks. So to think that you can just change something as complicated as a traumatic brain injury disease or a stroke disease by modifying one gene, it's nuts. And I'm saying that as a professor. I have the right to profess that. And it is my opinion, but it's not just my opinion. It's based on a lot of solid research showing that you have to think beyond the box. You have to think in a much more complex way if you're going to figure out effective treatments for many of these disorders. And again, one of the things that I want to say, there's a lot of other stuff out there, not from my laboratory. Right now there are close to 200 publications from 37 different laboratories using 22 different injury models in 4 species, all showing beneficial effects. That's a lot of corroboration. That is seriously a lot of independent corroboration, and it's very gratifying. That's the coin of the realm for people in science, that other people go out maybe thinking, we're going to show that this guy's a total jerk, and end up replicating your findings. That's how many papers are out there right now. And again, all these about mechanisms of what they found. Colleagues all over the world are now looking at this in a variety of other disorders. They're looking at it for the treatment of multiple sclerosis. They're looking at it for other types of diseases like ALS. Some people are considering it in combination therapies for Parkinson's disease, and on and on and on. So I want to just tell you now, where are we going from here? Because of all this work, the NIH of our country decided to support a phase 2 clinical trial. We're actually now in phase 3 clinical testing. A phase 3 clinical trial is basically the last step before something becomes standard of practice care. And thanks to our colleagues at the NIH, we are now in a nationwide clinical trial. And BHR, the reason I mentioned BHR earlier, they are supporting a trial that's involving every country in the European Union, including right here in the UK, Israel, Singapore, China, Russia, and Malaysia. So it's a big effort when you think about how many people are involved in a clinical trial. Just at the hospital where ours is done, it's probably 75 to 80 people that are involved in a clinical trial. You multiply that by close to 200 centers, and you practically have a stimulus package on your hands. So a lot of good stuff is going on, but I want to tell you about what we found. When we started out with this phase 2 clinical trial, which was just basically designed for safety and efficacy, this was a single-center trial involving 100 patients, we were basically told, find out the dose, see if there's any really bad side effects, and if you can, don't worry about it, but if you can find any signs of efficacy, that would really be nice. So remember I mentioned the window of treatment opportunity. We had to get informed consent from each and every patient or their family. And since most of these patients that were in the moderate to severe brain injury group, which is what we studied, 75% of the patients were in the severe brain injury group, 73% were males, by the way. So we had about 5 to 6 hours to get informed consent. So we couldn't begin treatment in this initial trial until we could get to this level. Now in the phase 3, the Food and Drug Administration has given us an exemption from informed consent, which allows us to treat and then get permission, which is very important because we know that the sooner you give treatment, the better. It's very unusual for the FDA in the United States to give a waiver of informed consent, but that's because of the data that we got. To summarize it in case I run out, in this trial, which was examined by a data safety monitoring board completely independent from the university, there was not a single serious adverse event due to treatment. That's pretty amazing. In close to 1,000 patients now treated across these 2 phase 3 trials, there have been no serious adverse events due to treatment. That's amazing when you think about a clinical trial. There have been lots of adverse events. People with severe brain injuries die, unfortunately, or they become seriously impaired. But no bad effects have been found that could be attributed by a number of different data safety monitoring boards to the treatment itself. So here's the statistics. I said about 71% male, 72.5% TBI. Most were injured in motor vehicle accidents, and so on and so forth. All members of the group had high levels of progesterone. We balanced for severity. And what I want to show you now is the data. When the data were broken, what we found is that normally, in the United States, people with severe traumatic brain injury, given basically state-of-the-art treatment, and all the patients in this clinical trial got state-of-the-art treatment. Nobody got denied anything. They were given either state-of-the-art plus progesterone or placebo, which looked just like the progesterone solution. On average, in the United States, about 33% of patients with severe traumatic brain injury will die. Probably because of the better care and better handling in the trial, the mortality rate in the patients given placebo plus state-of-the-art treatment had a 30% mortality rate. The patients given progesterone had a 13.6 mortality rate. That's pretty dramatic. We cut mortality by over 50% with just 3 days. Just 3 days of intravenous treatment beginning within 6 hours after the injury. When we looked at cause of death by neurosurgeons and neurologists, evaluated those patients who died due to other kinds of medical complications, heart failure, liver pathology, and what have you, as opposed to just those patients who had mortality that they would attribute just to damage to the head itself. In this case, 22% of the patients in the placebo group died, while only 6.5% of the patients in the progesterone group died. That's pretty exciting data. Nobody's come close to this. You look at disability rating scale scores. This is a battery of 5 different neuropsychological tests. To get to the point, what we found was that patients given progesterone had a fourfold better outcome than patients given placebo. So it's not just that we were saving lives and putting people into a persistent vegetative state. We're actually improving their functional outcomes measured 30 days later. Corroborating clinical findings. Shortly after we published our stuff, about a year later, a Chinese study came out, basically completely replicating what we found. They had 169 patients. They did them follow-ups for 3 months and 6 months. The NIH would not give us money to do any long-term follow-up because they didn't think the trial was going to work at all at this stage. And besides, they were only concerned with whether it was safe enough to give. Nobody expected the kind of efficacy that we saw in this trial. So they wouldn't fund any long-term follow-up, but the Chinese did. And basically, they had the same kind of lower mortality rates at 6 months follow-up, 18 versus 32%. They found, and just to sum it up, and this is all published again in Critical Care Medicine, you can see it, they basically replicated our results. They had a 32% mortality rate in their subjects. They were all severe brain injury. They didn't have any moderates. And 18% in the progesterone group. There's a very strong evidence, again, support for going forward, at least with a phase 3 clinical trial, which is where we are right now. We have we hope to test 1,200 patients. We are now in interim analysis. At 450 patients, there is an interim analysis going on to see whether it's having any bad effects, if it's having any good effects. There's the possibility that the trial could be stopped either way. It could be stopped because it's useless, if it would be futile to go forward, or it could be stopped because it's so effective that it would be morally indefensible to keep withholding a drug that has such a high safety profile as well as a potential for very high benefit. So this is a similar trial. It's both males and females 18 years or older with moderate to severe brain injury. It's a similar dosing regime, similar measures of outcome at 3 months, at 6 months, and a year follow-up. And that's going forward right now. I can't report to you the data because it's all blinded. Everything is completely blinded. We're not allowed to even get near the data. It's a completely separate group of people picked by the National Institutes of Health to evaluate everything. So what are remaining in the time I have left? We know that TBI is a very complicated, and stroke, a very complicated disorder. Our animal data for stroke are actually better than our animal data for TBI. We see marked reductions in stroke, infarct, and very substantial functional improvement. We're learning a lot about the mechanisms. Is a combination therapy better? Should we be combining progesterone with TPA, for example? Would we reduce the amount of TPA? The answer is probably yes, because progesterone, through its metabolite, allopregnanolone, upregulates endogenous TPA, so you'll probably be able to use less of it. And that means for higher safety if you get both neuroprotection and clot busting, and it probably will extend the window of treatment for TPA. As we've seen the progesterone, we know it can be effective up to 9 hours in stroke. There's no question it's highly effective even after a delay of 6. So, can it be used in other neurodegenerative disorders? A lot of people in Italy are looking at it for treatment of multiple sclerosis. Why? Because when women with MS become pregnant, their symptoms virtually disappear. When they give birth and progesterone levels drop, they get a marked exacerbation of their symptoms. What did I tell you earlier? When a woman is pregnant, her progesterone levels are maybe 50 times higher than normal. And I think, and because progesterone stimulates both glial cells and Schwann cells in the peripheral nervous system to make more myelin, you may be rebuilding the myelin that leads to the amelioration of symptoms. But this may require much more chronic treatment than the kinds of treatments we're given. Because in the TBI and stroke, we're not giving more than 3 to 5 days of treatment. That's what's so amazing about this drug. And of concern to some people in the pharmaceutical industry, because it doesn't have to be given over the lifespan of the individual. It only has to be given early in treatment. And this is problematic if you're looking at this from an economic perspective. So where else are we going? Our United States military, obviously with 250,000 brain-injured patients, is very much interested in knowing whether we can get this into field treatment. We are doing that. We've made remarkable progress working with our colleagues in chemistry at Emory to develop a far-forward field treatment to make progesterone much more stable, much more soluble, and available in a form that any EMT technician, any fireman, any police officer, could carry it and give it under the without having to do anything more than give it an injection just about anywhere, whether in the arm, the leg. Sometimes when you're dealing with very severely injured patients, you can't just give them an inhalant. You can't just give them, an intravenous or stereotoxic implant. You need to give them something fast to get started before they even get to hospital to do as much neuroprotection as possible. So that's kind of the end of my talk. I know I went into my New York City speaking mode. I went very quickly, but I want to write this down. My website, my new website, is www.brainresearchlab.weebly.com. And if you go on there, you'll find all the information, the publications, the TVs about patients who've recovered, all kinds of interesting things, including many articles, and a lot of stories about the people in the lab, including Justice. So take a look at it and see what you think. Did I finish on time?
That was fascinating. You've done extremely well because we were sort of talking very fast because we started a bit early. So we've time for 1 or 2 questions. Please tell us who you are. So have we got a microphone or are you just going to shout?
I can talk loud, it's okay.
Okay, that's good. So we haven't got a microphone. Okay, just talk loudly. We can hear you.
I'm Melissa Shapiro. I'm a PhD student here. But I was curious, you said that if progesterone is so evolutionarily effective, why do levels only surge during pregnancy? Is there some bodily cost for having high levels of progesterone weight, even though there's great protective factors?
No, you get high levels of progesterone also in your normal cycling. In your luteal phase of your hormonal cycle, it will also go up. But it's not sustained for the 9 months that it's sustained once you become pregnant. So it's always going up and down. Remember, I mentioned that we damaged our rats during the luteal phase of their cycle when they're high in progesterone as opposed to high in estrogen. The rats cycle about every 6 to 10 days. So you could look at it makes it much easier to work in those animals.
But why isn't it high all the time if it's so protective?
That's a why question. That's an interesting, I can't answer a why question. That's a teleological question. I don't know the answer to why. We haven't evolved for that purpose where you're constantly in a state, because I think if it stayed high, well, one thing for sure, if it stayed that high, you'd never become pregnant. It would block ovulation. So that's probably why. But that's my guess. You'd have to talk to God.
Well, God's coming later, so we can ask him or her. Yeah.
Hi, I'm Ryan Vesky, I'm a first year undergraduate Medicare. I was wondering, did you look at any of the women who are possibly on the combined oral contraceptive before they had the traumatic brain injury, and if that affected in the placebo group their recovery?
Right. The question was, did we look at any women on contraceptives and do they do better? We haven't done that because, as you saw, most of the people in the traumatic brain injury trial, 71% were men. Now they're actually, with 1,000 patients, this is part of the stratification of the trial, to go back when the data are made publicly available to look at whether being on contraceptive, being pregnant, or any of these other variables, or how, because we're not limiting the upper end. We've had patients in the clinical trial. This was a mistake, I think, but that we thought about excluding patients under 18 and small children that weren't even in cycle. But we never thought about the other one. So we've got patients as old as 90 years of age in the clinical trial. But so once the data are out, then those data will be available. They are stratified, and we will know more about that.
Any more questions? Can you say a word about what's the natural history of people with traumatic brain injury? What kind of happens to them over the years?
Depends on the severity of the disease. It's been reported in the literature that people with early brain injury from concussion, well, if you look at the data coming out of the National Football League, how many people know what that is? Okay, not too many. The National Football League is our major professional football players, American football, not rugby. And it's been shown now that there's a chronic encephalopathy that develops as a result of repetitive head injuries. So a lot of football players that are in their 40s and 50s are developing early onset dementia as a result of these repetitive concussions. Now, it's not just because they're professional football players. You've got to remember that most of these guys have started out when they're 8, 9, 10 years of age. And they're always in practice as well. When they're not playing the games, the 30 games a year that they play, or 15 games a year, they're also in practice, preseason practice, and constantly getting those hits. And now you just read that in the NFL, there's a bonus. You were getting paid a bonus to go out and hurt your team opponents. They're paying them big bonuses, $50,000 to go and hurt the tight end or I forgot the name of the ball guy. I'm sorry, I'm embarrassed. But so it's a big issue. And now with the PTSD, the post-traumatic stress disorder, they're learning that months or even years after the injury, that people can develop these symptoms of extreme depression and violence and other mood disorders that are directly related to the injuries that they had. And boxing, of course, a lot of people develop Parkinson's disease, professional boxers. So there are a lot of pathologies that emerge years out after the initial injuries occur.
Good. Okay. Thank you very much.